So thank you, Sarah, for the kind words. And I'm Shruti Bhargava. I'm a postdoctoral researcher at University of California, San Diego, and I'm working with Dr. Jason Sicklick. I'm going to talk about my project, which revolves around establishment of patient-derived succinate dehydrogenase deficient GIST models for predicting the therapeutic response. Now, as we were going through the introduction of GIST for previous few days, I'll just give a very, very basic refresher. Now, we know that GIST or gastrointestinal stromal tumors are predominantly present in stomach and are present in the wall of the stomach. Secondly, the cell of origin of GIST is thought to be ICCs or interstitial cells of Cajal, named after a famous neurophysician or neuropathologist, Cajal. It is also characterized by the expression of characteristic markers like KIT and DOG1, also known as ANO1 and TMEM16A. These markers are actually membrane proteins and are present in high amounts on the GIST cells. Now, next we go to the genomic alterations that are present in the GIST tumors. There are a plethora of alterations that are associated with GIST tumors. And to name a few, we have been discussing about KIT mutations as well as PDGFRA mutations. Yes, the proportion of GIST that are having these mutations is pretty high, but we also have tumors which have the alterations in other genes, especially the SDH genes. That will be the focus of my talk for today. Now, what is SDH? SDH is succinate dehydrogenase, and it is an enzyme which is present in one of the organelles, which I'll be talking about in my next few slides. And when we go as the anatomy of the tumors, we do find that these tumors, SDH deficient tumors, are mostly present at the distal end of the stomach, whereas the other tumors are also present at other, like the antrum or the beginning of the, fundus or the beginning of the stomach. Now, what is succinate dehydrogenase enzyme? It is present in one of the very important organelle of the cell, which is mitochondria. It's also the powerhouse and is responsible for energy generation of the cell for various processes inside a cell. The complex consists of 4 subunits, which is SDHA, B, C, and D. Now, the main process that is catalyzed by this complex is the conversion of succinate, that is a metabolite in the cell, to fumarate. So the product of this reaction is fumarate, whereas the substrate that is being converted to fumarate is succinate. There can be various ways by which the succinate dehydrogenase can be inactivated in a cell, which can then lead to the GIST formation. Now, I'll be talking about 2 of these. One is the mutation that is present in either of the 4 subunits that are there. And these are germline mutations. They can be passed down generations, that is, they are hereditary, and are often found in the Carney-Stratakis syndrome, wherein we also find the presence of paraganglioma along with GIST in these patients. Now, another mechanism by which the complex may not work in the tumors is by epigenetic modifications. To simplify, the gene that is present may not be expressed because of certain modifications that are present upstream to the gene, which causes the protein or the subunit to not be there in the cell. So say, for example, if we are seeing the SDHC gene is repressed or the RNA expression is low, then the protein will not be there in the cell, making the complex fall apart, and then there will not be a functional SDH complex, leading to the changes that are downstream, which is mitochondrial dysfunction, abnormal respiration, and thus cascade of events that happen in the cell. Now, the classic or one of the challenges that we are facing clinically in the SDH deficient GIS is that they're having very modest response rate to the available tyrosine kinase inhibitors, which are the first line of drugs in the KIT mutant GIS. So the present challenges in the field are we don't have, like until recently, we didn't have good patient-derived tumor models to understand the biology, what exactly is happening in our disease, and also to screen drugs to make the therapeutic options available for patient care. So we picked up a basic strategy to meet these two challenges. Firstly, to establish patient-derived models, we dissociated the resected tumors, and then in a cocktail of a very stringent permutation combination of ECM, that is extracellular matrix, and media were tested. And then we found an app combination which can actually sustain the growth of these tumor cells outside a human body. We also now, next challenge was to see whether whatever we are growing in the dish, is it actually similar to the parent tumors, or is it getting adapted to the external environmental conditions that we are providing? So that is what we compared with different tests that we performed. So we, in our research, had taken representative tumors from SDHA mutated tumor, SDHB mutated tumor, and SDHC mutated tumor. And then we had established the cells in ex vivo in a tumor dish where they can grow both as a 2D layer, which is now termed as adherent culture, and they can also be cultured in a very floating condition, which is now termed as non-adherent condition. Now we check for various characteristic GIST phenotypes, which are specifically present in the SDH deficient GIST to be present in our models that we have just established. Firstly, we check for the expression of the characteristic markers that I had described, which is KIT and DOG1. And we do see that KIT, which is present in red, and DOG1, which is present in green, is expressed in the parental tumors as well as in the adherent and non-adherent tumor models that we have established. Now, the second characteristic feature of these tumors or tumor models was loss of SDHP subunit from all the 4 subunits that we have talked about. Now, this is a very unique feature that is present in SDH mutated or SDH deficient tumors. And we do see a loss that is present in the SDH mutated cell models that we have established as compared to a KIT mutated tumor cell line. We do see a replicated result in the immunofluorescence assays as well, where the green in staining, which is the SDHB protein, is lost in the cellular models that we have established as compared to a KIT mutated GIST cell line. Now, going into a little bit of detail about how SDH is working in the cell, as we were discussing earlier, SDH enzyme complex is formed by 4 subunits, SDHA, B, C, and D. Either of the mutations in any of these subunits can lead to a defective complex and thus abrogate its functioning. So say if the function is abrogated for the SDH complex, we will have a lot of succinate in the cell and a little bit of fumarate in the cell because the forward reaction will not be catalyzed. This was also seen in our results where we had higher levels of succinate in the cell in SDH deficient models as compared to the KIT mutated cell line, and the fumarate levels were lower as compared to the succinate levels in the cell. We also saw a deficient respiration in the SDH deficient GIST models as compared to the KIT mutated model. Now, so far, we have seen that the SDH complex is playing an essential role in the TCA cycle or the cellular respiration. It is converting succinate to fumarate And with its abrogation, we have higher levels of succinate in the mitochondria and inside the cell, and lower levels of fumarate. So what are the other things that are changing inside the cell, which can be then driving the progression of these SDH deficient GIS? We do know from the literature that higher levels of succinate can have various other things that can be like malfunction because of the presence of higher levels of succinate. And one of the essential organelle in the cell that will be affected by this increased succinate levels is the nucleus. Now, we'll see in future slides how that is affected. Now, the other thing that we have characterized in these tumors is transcriptome. As I was just talking, nucleus is the region where DNA is present, and that is now transcribed into RNA, which is then going into proteins, which are like the front workers in the cell. And we were checking as to how the transcriptome is present inside the parent tumors, and is it reflected like reliably in our just mutant, SDH mutant just models that we have established. And you can see high correlation ratios that we obtained, which essentially suggests that the SDH models that we have established are fairly similar to the parent tumors that they are derived from. We also saw the gene expression signature that is associated with SDH loss to be highly present in the parent tumors, as well as the established cell models, as compared to the KIT mutant cell line. Now, another feature of these tumors is that they mimic something in the cell when it is deprived of all oxygen, and that state is known as hypoxia. This state is generally present when there are lower levels of oxygen externally, but then due to high levels of succinate in the cell, it almost mimics a similar cellular state where the HIF1 alpha, which is a sensor of oxygen in the cell, is stabilized, and you have higher concentration of HIF1 alpha in the cells. So succinate actually inhibits one of the components in the cell, which causes the degradation of this HIF1 alpha. Thus, higher succinate in the cell will have higher HIF1 alpha, which will then cause higher levels of genes which are dependent on this HIF1 alpha. So we did see that in our SDH deficient tumor models, indeed, there was a high level of both HIF1 as well as HIF2, both of which are actually associated with hypoxia or with high levels of succinate in the cell as compared to the KIT mutant cell line. Now, this was also translated into the gene expression that is associated with hypoxic related state. And we did verify that these are actually higher in the SDH deficient models that we have established and the parental tumors from which they are derived from as compared to the KIT mutant cell line. We also verified our result by performing quantitative real-time PCR, and we did see that target of HIF1 alpha or HIF2 alpha like VEGF-A are higher in SDH deficient GIS as compared to the KIT mutant cell line. So clinically, we had discussed that SDH deficient GIS models are actually refractory to the already available tyrosine kinase inhibitors like imatinib and sunitinib. This particular process is also recapitulated in our cellular models. So our models not only mimic the molecular characteristics of SDH deficient GIS, but are also now validating the clinical aspects of the SDH deficient tumors, like therapeutic resistance that is already present. So the data that I'm not showing here is that the KIT mutant cell lines were showing lower concentrations of imatinib and sunitinib to be effective for cell kill, which is depicted by the slope of the curves here, whereas the SDH cell lines were really, which required more concentration of the drugs to bring the same killing of the cells. Now, one of the drugs that we found to be effective in these SDH deficient GIS was temozolomide. That is a DNA alkylating agent, and it is known to intercalate with DNA, cause DNA damage, and thus lead to cell death. So we did find that all the three GIST cell SDH deficient GIST models, which is mutated either in subunit A, B, or C, are actually quite sensitive to temozolomide. We also then asked a question, is this due to the loss of succinate dehydrogenase complex in the cell? So for this, we collaborated with researchers in OHSU, and the SDH gene was knocked out from the cell. So here, the black line denotes the cell line which has active or normal SDHA. The red here denotes the line which is now having, which does not have SDHA. We also did a rescue experiment where we overexpressed the SDHA subunit again in the deficient cell line. So if you see the therapeutic response to temozolomide, the wild type cells are mostly refractory to temozolomide. Then when we do a knockout, they become sensitive. But when we place the gene back again and the complex is functional, it again becomes refractory to temozolomide. So indeed, the temozolomide sensitivity is associated with the functional SDH complex. We also reciprocated the result with another assay, which is called formation assay. And here the purple intensity is associated with the cells that are present actively. So if you see empty wells, it just means that there is a higher cell killing. So knockout cells were killed faster and at a lower concentration as compared to the wild type or the rescue cells. Now, as I just discussed, temozolomide is known to induce DNA damage and double-stranded DNA breaks. It is widely used in other cancers like glioblastoma, melanoma, and thus the mechanism of action is available. So we also wanted to check if in our cells it is the primary mechanism of action, or is it also inducing something else in parallel with the DNA damage? We did find that the DNA damage was happening, and we performed a comet assay wherein the comet or the tail length of the comet is indicative of DNA damage. Longer the tail length, more the DNA damage. So if you see temozolomide caused an increase in tail length, which is indicative of higher DNA damage when the cells are treated with temozolomide. We also checked in another way if DNA damage or double-stranded breaks are already happening in the cell when temozolomide is present. And we utilized a DNA damage marker. Now here in this assay, the more proportion of cells which are having the red intensity or red staining, that is indicative of more cells which are showing a staining of gamma H2AX, the DNA damage marker, and thus it suggests that there is more DNA damage inside the cell. So we do find that when the cells are having temozolomide treatment, there is a higher proportion of cells which are stained with gamma H2AX to indicate that there is a higher proportion of DNA damage occurring in the cells which are now exposed to temozolomide. Now, the most important question of our entire research was whether all of this is now translated inside the clinic. Is this finding clinically relevant? So we saw that when a patient who was SDHB mutant, who was having SDHB mutant GIS, was treated with temozolomide, within 8 months, we had a dramatic response. And thus, the phase 1 clinical trial of temozolomide on GIST patients gave us promising results. We also saw that there was either a partial response. Out of the 5 patients who were treated, 2 were giving partial response, whereas others were giving a stable disease. As is also evident here in the swimmer's plot, where we do see that 2 patients were having a partial response, whereas others were showing a stable disease. And I just want to point out here that the patients were coming from SDHA mutant GIS, SDHB mutant GIS, SDHC mutant, as well as SDHD mutant GIS. Now, to summarize our findings, we found that the SDH, we actually were able to, for the first time, establish reliable patient-derived models that can be used to understand the biology of the tumor, as well as to have drug screens performed in them to find more therapeutic viable options for these patients. We did find that when the SDH complex is inhibited in the cell, there is higher levels of succinate and lower levels of fumarate in the cell. The increased levels of succinate then gets translated into different alterations that are associated with these tumors, like increased HIF1 alpha dependent transcription, leading to increased proteins which are present normally in a hypoxic state in a cell, which is like mostly present when lower levels of oxygen are present. It is also associated with higher DNA methylation, which is the data that I've not shown, but we've already validated that there is a higher level of DNA methylation present in these cells as compared to KIT mutant cells. On a clinical side, we found that temozolomide is effective in these SDH deficient GIS, both in vitro as well as on our GIS that we have treated, GIS patients whom we have treated. It causes an increased DNA damage inside the cells, and that leads to cell death. So because of the promising results in phase 1 clinical trial, we developed a phase 2 clinical trial with temozolomide in advanced succinate dehydrogenase mutant or deficient GIS, which is currently ongoing. And it is open at UC San Diego, Oregon Health and Science University, Fox Chase Cancer Center, and University of Miami. Our target accrual aim is 23 patients, and to date, 13 patients have been treated. Now, with this, I would like to thank all the members of Cyclic Lab, then our collaborators from Salk and from all the other universities who have contributed immensely in this study. We also thank deeply our funding agencies, the advocates and the patients for constant support and encouragement, without which this study would not have been possible. It motivates us daily to have your support and encouragement. Well, thank you so much.
Thank you. Thank you so much, Shruti. This was an excellent presentation, and it definitely gives us hope and encouragement of what research is being done on SDH deficient GIST. So you may have really touched on this, but I'm just going to ask it again, and maybe you can also think about with some of your colleagues how they may answer this. You know, as advocates, we want to see what else can we do to help contribute to SDH deficient research. And so the question is, what do you think research scientists need, and how can we help that to help move research forward?
So that is actually a great question. And this has been just discussed earlier this morning. Even I feel that collaboration is really important. First and foremost, we all know that SDH deficient GIS is even rarer than other cancers as well as other GIS. So we do need collaborations where we can just come together with different expertise and thus do well in the fields. There is not only one aspect that we are looking at when we are studying the disease, but we are looking at the disease from different aspects. And that is like really important when collaborative efforts are put in. Like we currently have physicians, computational biologists, wet lab scientists who are working on the field. But even in that, if we have diversified expertise, as well as for the research material, it'll be great if we collaborate and thus facilitate the research in the field.
Excellent. I mean, not to put in a shameless plug, but the SDH consortium does help
Yes.
pull different stakeholders together to help collaborate. You know, I know that sometimes we have advocates in Europe who want to contribute, but sometimes it's hard with transferring tissue or especially, you know, fresh tissue. Can you think of maybe some other action steps, even from other global sections, that could help contribute to providing more centralized collaboration to advance the research in the field?
Yes, for sure. So we do need, like, for example, I just showed a few of the slides where gene expression data was mentioned. Now, if we do have that in a collaborative effort from all across the globe, then we'll also be like having representation of how the patients are having variations in the gene expression from different regions. Is the race and ethnicity having any contribution in differences? Can that be translated into a personalized medicine for different patients? So this is something I feel that will be relevant. And if we can have collaborations all across, it'll be great to have such a consortium, which will be really informative, and then will then just facilitate and personalize the medicine that we are delivering to the patients.
Excellent. Okay, good. There was a question from the last session regarding some other agents, specifically the, let me scroll all the way up, some immune combination therapies that gave a durable response in wild type GIST. I know that this wasn't your presentation, but I was wondering, as you do test drugs with those cell lines, have you seen any sort of immune agents or combinations that may look promising that you could share with us today?
So that's, again, a very interesting question. We haven't directly tested any immune agent, but we do feel, because the SDH deficient GIST, at least at the primary site, which is stomach, is not having a lot of immune response because the immune cells are not like heightened in their response at the primary site. So are there ways where we can actually activate or like recruit more of immune cells to the primary site is something that we need to think about in the field. Also, we all know that SDH deficient GIST is metastatic. So it is present in different sites like liver, lymph nodes, and other sites. So is it that different sites are having differential response to therapies? Is again something that we don't know. So there is a lot that we don't know, but we would like to know. So that is a reason we need to have like collaborative efforts and then just like go through each and everything.
When an SDH deficient patient has progression, have you seen that it mutates into other types of mutations, especially when you're saying then there's like growth in the liver? Have there been biopsies to show that that mutation is different?
Again, very interesting, but we don't know the answer for that.
Okay.
So in other cancers, it is known that it is the case that there are different clones that may be present in a tumor, and some of it can be overrepresented at other metastatic sites with similar molecular aberrations or with different molecular aberrations. So that is something that we don't know about SDH deficient GIST, and we would like to know if that is the case here as well.
Okay, so everybody listening today, I think it's important if you've identified patients with wild type GIST, SDH deficient GIST, that you come forward and find a way to share this information with this network, because it helps us really discover more clues about the mechanism of this disease. Any other questions before we conclude this session? Okay, I think we're good. I want to thank you so much for joining us today. Always a pleasure, and we learned so much. Thank you very much for presenting.